We can start. Yeah.
Great. It sounds like we're ready. So good morning, everyone. Shukpatale. It's really wonderful to see you all again today. I'm excited that you all are back. Yesterday, we spent some time learning about plant growth and development, and today our main focus is going to be staying on that topic of development, and today we're going to shift our attention from plants into looking more at animals and especially looking at some examples with human development.
Cerchitan Sepre Coladus Tires, the Tirimbana Sukaritagin Sena, Joshiti, and the Cerchitan Serpegon and Labata Serpegon and Debakanshila, Tirangi Joshiti to Sochache, Sochan and Muskerji that and drew Mila Pisha de and Eco Serpeti Kandajungo medical ties.
And of course, we are continuing learning some teaching techniques. And you can see that in the first learning objective for this morning, saying that we are going to brainstorm some examples of teaching using visual representations and models.
Janayagi Hunsente and a visual representation system, but like a removal like Kanasuko, you may last for a while or take Talayan.
And then after that, this morning, we are going to go through two different ways of looking at embryonic development. And they are both two correct ways. It's just there's a lot going on in embryonic development. So it can be helpful to kind of look at it from a few different perspectives so that eventually we get the whole picture.
Shopa so the Tompatica Simbanga to Drumsang gets her pregocolaya Kusugia, Samlutantani or S, Roarco, Pizuma Sona, Nipechetan, the Tandashijagores, or Tini Chalanga dus Chonder Chichitagi, that take up to Carrioresena, Chetangi, the Drumsang gets her pregamatelaya go. Nita Mampuch and Jurandugo Yozans, Tonto Mampuch in Jungo Yozan, and the Telaya, and Chicho Mando and Taba in Baina, Yangasulaya, Kugua de Kanza Chilaya, Pentocres.
In our lesson yesterday, we already got to see some really good examples of using visual representations in learning. And this was happening as you all did your drawings of the arrangement of the water molecules as a solid, as a liquid, and a gas. And then also you did your drawings of the, if ice were to be more dense than water, what would happen in the ocean. So we already have some really good examples of those drawings.
Tambotelaya, and a chugin dudig copa candadochi, mentor mula copata, and a chugin duduti cook chugin number sumging walla da picosura, Sherzutan Sazutan, Lanzu Sumbula depi, Capsa Candado, Pagatecha sugar mula coach, Tayore, Nanshingi, and Katie, and a chapagi to say the chuli. Lichua chapi capsa, and Nathan Canado which into Gresa, the answer to Remula coach, Mukatoy, Chugi, Armula coach, dus Janyores.
Drawing is not the only kind of visual representation. So now I would like to ask you all in each of your groups to brainstorm and make a list of some other kinds of visual representations. And I would encourage you with these, if you can, try to be as specific as possible. So if you can think of maybe a specific example that you have used of a visual representation in a course that you have taught, or if there's another class that you have taken where you actually got to experience this visual representation, that would be really helpful. So if you can get as specific as you can with these, that would be really helpful.
Tanda Chesulaya, nature trader Chiagi, do Seguchitagi, yes, Tekare system balai, Matsugu, make it why you Pitsun Pechetanga de Dansagura, the Tonrong Pitsun Pechetanga de Tanzanta Tragora, Tonrongi Chegulatani de Nenzana Tragora. That this pickups a Tonrongi Chegutan, Tonronga Tablantan, this pickups a remote Triatichipo Mares. shenda mampo yores ta shenda mampo yobate ta kare kare chi sare chesu khana la tu de chiro ches chi chi ba na chorang yang zinda shenda trip up se ya chik mu ge thwa yo chu ge te la ten visual representation se gwa mi ki thwa yo chu ngo la ten chi de tan zinda thia susugin zinda trip up se peche ta ta yang na susugin zinda chi la ya zushi chi pick up se yang gela agenda ge ya tena do peche tang yo ba la so pa gang la ya du street na ro ches.
Yeah, so go ahead, take a few minutes, and then we'll share with the group.
Ta kama khashi na la ya te ko la street na ro ches. Nti jela nga sunyam the street na is.
And we're going to start to come back together.
That's all to china sta. Ta ya sama ya zero che. Nyam to the chain da.
I would like to hear from each of the groups, and I like to switch up the order a little bit so that it's not always the same group that goes first and the same group that goes last. So I think we'll actually start in the middle today and then swoop around like this. So the middle table, could you share with us one of your ideas?
Ta ti ins ro ta da ro kha su ngone kare tu de chwa te son ro ches ta tere ba na ya check-in zinda traffic ka sir wa
Hello. Nga so ro kha ge de le ta chik lan do ra ta da graph reach do de na la bar graph da histogram da line graph thone de
using graph. Yes.
chik ran la
Yes. Bar graph,
histogram, line graph, extra.
Pie chart, all sort of graph. You want to go to the last one?
Yes, we'll go then to this table here, and then I'll go around. So this table like with Gaelic.
Gele chechu ngone. Re son ro che kura jandwa insa.
ane ngazu rukana chida ngatuk to the dina chik shuna ane mito sashe demonstration there is.
Demonstration.
Okay, do you have like a specific example? We can write demonstration here, but do you have maybe like an example of a demonstration that you've seen before that you thought was really good or really helpful?
Demonstration sana tanda ge ti kari imbes.
ngazu gi ane jeans tang dende ane pu den zo yochip ne kunzu so chue do tap chue de ya
Thap chiga tere zo che.
tap chu na de do yang ngazu kunzu la di chalak do ten yang chik so chu na kare na de
Ta so te gela ge dus te tirwa.
las
Ra. Te tirwa gela dus. So he's in the case of a demonstration, he's talking about two cases. One is role play. You can have someone to role play that roles, and also, for instance, like creating the double helix structure. Teacher can show in front of the class how that is made, and then ask the student to make the yes, to show in the class how to make the model making. Yes.
We'll come here.
Educational trip. Yes. Or field trip, right?
That's a good one.
That's you're it.
ngato dinu tanda tamobuji do tenole tanda body language zubogo no jigten try
Body language.
Oh yeah, so I think that makes me think of two things. I mean, maybe even like, I don't know if it's exactly body language, like, but what the two of you did yesterday with the atom. Are you thinking of something like that? Like the cell phone?
Zubugan just sebu kapse kesan gelagi kasa.
The electron.
Changwa kola system bala ya ko balance bound kola system bala chigidwa. Tase ko yorwe.
I would even say like acting, you know, that
Yeah.
type of body language.
Acting, yes. Or role play, acting, yes.
We'll go here.
Yeah.
마수기 소소기 조치야죠 알라바히 그냥 빠가노든데 키워니 잠바노든데 키워서 이름은 비즈데 조치야다 도도
Tade gala yo chugo model making saitri doara.
아리
Aha.
디즈노 다소 조에티 라바가 조에티
So using use Play-Doh sort of stuff and making the models. So model making is
Like 3D
yeah. Right.
crafts.
So using like a Play-Doh to make structures or the cell. Wait a minute.
Yeah, great. So that's a really good idea for like the specific way to do model making. You can do it with Play-Doh, you can do it with clay. There's different kinds of materials to work with.
Clay or samba or
Yeah, samba. And then you can eat it.
yeah. Teaching ma tension kandu. Teaching ma tension kandu. Yeah.
Is this one not good?
Yeah, it's too loud or
Oh, I
you
can. I mean,
use
if it's too loud, I
too
can.
much,
Just,
है ना?
if
Much
I just
tension
move it
kan
away, is this okay?
Yeah, that's okay.
Okay.
Yeah.
Yeah, it's every time when I put it on and take it off that it moves.
Yeah, that college. Cheso college.
나좀 거리가 있는데
Ah.
거리가 있는데 때라
Cái nhện sẽ là ta có cartoon tiền là thế.
Oh.
So movie tiền sẽ có à?
애니메이션
Animation. Show you animations. Yes.
Digital animation or
Digital. Yeah, digital.
At the back
Yeah,
corner
tene.
now.
Jalo.
저데 직소 디오 루트게 좋아다 세르다 로가로가상 수 루트 직소 런지
Jigsaw activity.
Yes, the jigsaw. I love the jigsaw. So just very quickly, I think most of you probably have done jigsaw, but maybe this table could also just give a brief explanation of what that means. Like, what do you do in a jigsaw?
Tada de ludik chegu system bala ya kari chegu language. Teach sonro chidas chita gone. Chichibane chesu ludik chegu sena kanda imbate haku yo chires. Chesu ngoni tanda ludik chegu se bikapse kari kugu yo me teach sonro chesu.
셀기 나로 차시에 미라미라 여러와 디게디 네 로가샤네 부구수소 디주야다 네 가리나 용구라 맞대야 된대 용디 대시에 나래 가리나 셀기 차시에 여러와 로가로가서 안수 루디해 줘다 루디네 맞대야 된대
For instance, we can have the part of cells and apart, and then we can ask the student to put them together to form the cell.
Oh, okay. So yeah, I was even thinking of a different kind of jigsaw, but yeah, definitely that's that's one that could be one kind of jigsaw, and that's a really good example of a specific activity.
They ko tanda ludik chegu system bala ngi samle jenda chikorde sha simbana tanda chezo songatila yagudus ko chegu na shabana yagudus. Tagoles.
Great. And then yeah, back middle table.
nganzode shu thermocol bijudanle jijingi ko basyo jora odiche
Kari peche tanche?
thermocol na ngad chala parolo shu ti sosoro shulo isto odi bijudan selgi ko bat structure dise
Kasa ko de la ya thermo like that.
thermocol na chala ko madu bodo shu soto chido odi
So making models using the thermocol. Do you call thermocol that what do you call like
Oh, I think is it styrofoam? Is this what we
styrofoam?
talked
Yes,
about?
yes, styrofoam.
Okay.
Yes.
Yeah.
yeah, using styrofoam to make models. Like making a model of cell.
All right, and then you have a challenging task since we already have 8 ideas there, but our final two.
Tamate cheso.
네 만소 마무지가 그때 발치하시 잘서 옛날 당한수 아니 전통 지식 곳에도 내 만소 참봉이 와라 참봉 거니 다 중국 바바다 변나 참봉도서 간지지 요미도서 못지
타동 70페이지 땅에 딱 차풍고 고발라다 들어와.
어디래
아하. Using a microscope to see the structure of the cell.
That's a great visual that we also just did yesterday. So yeah, that's great example. Yeah, thank you for this list. And I wanted to put it up here so that, so you've already seen that we used several of these kinds of visuals in class yesterday. And now that we have this list there, thank you for writing the list, we can keep an eye out for which of these kinds of visuals you'll notice in today's class and even in the coming classes throughout the rest of the week.
Hada dandagi thude pe apuna sons thude du shashagi yes ro dan digna ne khasichi imbaina ngasu khasan jiore khasichi imbaina tiran jinda khore tena ngasu ge jingi gin zandan de traffic dantan de na la yang ko khasi pechu tangre tis tambala ya tambadela ya chi tona chigi na ro che sta de ngasu ge miki thorong ge ane tablen tin pechu tanga 들어와.
Here, I wanted to just share a few benefits of using some of these visual models. You may have thought of these at some point in time, but it's still just to let us know that there are a lot of different benefits to these.
Tada dire kasanga su thorong ke pichun ti peche tamba imba na khipen kare yore se chinde se jagis chirgi imba na ta khipen mambo yore swa dus chi ba imba na thorong ke pichun ti peche tamba imba na thon tu bi pichu peche ta chin chin da ba imba na dan de la ya ku khipen khashi chi dus ngu chu yes.
The first one is that it can help to activate attention. So even so far today, there's been several visuals. So you can imagine it would be a little different if I were to just stand here, talk to you, nothing to look at, you only had words. It's very helpful for our attention to have some combination of hearing some words and seeing some visuals, and then sometimes even some other senses used as well.
That tampote kare system balaya ane ko tuno chela pengres tam ko sopti ki tuno tek chigala pengi ores ta pisha shapa imbaina ta tanda de la ya ngasu ge yang ko mi ke thwa yo chu ge ta pichun khashi ching du special tang de tan zenda trigi ores ta ke tin de la tesh ga me chu de la tres ga me chu che chechu gen de lo ne ngarang ge zenda te pe te kha ge chang she chi thi bai bai na ant ko ne ta chi cha gro ati kap se ko tuno me ba ta ko tuna de la duwa ge ne ta ta yong gro as is ta bala ya ta de che bai bai na ko mi ki thwa yo chu de che bai bai na te ke I ta drati keti kuatsa maimba ikite miki thungu yora ikita remula sopo miki thungi kapse imbaina so tizu ge tona te tangwan de la yila che pasukura ta te tono te kichi de ala pengres.
They can also help to activate and help us remember and connect with some of our prior knowledge. Sometimes when you see, even if it's a new picture, a new image you've never seen before, you might just kind of remember a little bit of, oh, this looks similar but a little bit different from something I saw before. And so then you might get curious and ask some questions about what is going on that makes it a little bit different.
Ta ya shendo chi kare chi gre system bala ya chi per tang nya la so po thong bai bai na ya su su ge ngon chi xia ge su su ge xi xia da yon de ti ya ten su ya te chi gre ta ti kan ro re se na chi Tering ta tanda rang tenju ko perti ngomba thonyo me pai bai na ya ngomba thonu ko perti ta tis chanda yo pai na ale de ngomba thoya ke perti ta tis ki daw dus ta tila ya khepa kari chi gona de la drewa kari chi gona asle drewa tus ko pala ten de ti ro tila ten yo ko shisha saba jang ge ko ka tu yung yo res.
For reducing cognitive load, really this just means that the visuals can help make it easier to understand. And you'll see as we go through our lesson today that especially for this topic, if I were to try and only use words to describe this to you, there's no way. We would, all of us, myself included, be so confused. So all of the visuals will really help it, make it more understandable.
Da thorong ke pisan shepa yamba na shenda chi kare tangare kare pen thokare se na ta nginjing gi khu jinyon tangare se de kar se na ta ngasu samlu chembu tanga de ta jang gwa chi gorwa otte chi gwa ke ga mares ta lapar ta tireng ge joshi de ta ucham bai bai na ane chi rumu ta ta nya ta la supan di me chi Sixty Pugan, Triguyo Pambana, Chetu Gutongre, Ngayan Gutongres, Karuchi like you make Kasuchagamares, that dick of Simbana, Hermutan Tambai, the Remuta Perla, Agili Tripambana, Nichi Kuala and Lilautan, and some Lushi Chambutama Gugan, Cotonia, or what the young.
for building the mental models, it's really helpful to have an image that we see, on the screen or in a book, and it just helps us create the image and the model that we have in our own mind. So that's another benefit that can come from that.
다 젠다 카르시스템 발라야 수수가 삼나라야 지 비다 비춘지 철기지가라 팽그레스와 다 비차샤바는 데라 테나야게 피르탄 르몰라 소파이나레 체수고 록테나나 요페게 피르탄 르몰라 소파이레 तेतला तेन तेतो थोला तेन देते ने जेला या ती कोला जोशी ती का सामलो तं पायना सामलो ना तेले छेगा चियों ग्रेस अले देते दिनदा चियन से छेगा चियाला फेंगियो रेस.
And finally, they can help support transfer of learning. And so just what do we mean by transfer of learning? One example of this could be that you've probably talked about atoms in your physics classes. And then yesterday, we were also talking about atoms, but in a little bit of a different setting in the context of photosynthesis. So the images can help with this, that transfer of learning from similar topic that you learned about, but maybe in different classes.
다 타마틀라야 나와데 카레시스템 발라야 수수고 장샤주고 셰셰치 셰셰 타코 딘신 찌니 딘신 젠다 치라야 파슬라야 타치 키알라스 고로 분절당야라야 팽그레스 타 피샤샤나 커스고레스나 다 케사 찌르게 만나체주고 도탱고 코르테 네치 항윤컴 세르코네 장 요스 바레스와 항윤컴 세르코네 코 도탱 장바임바나 아니 개성아 속에 영권이 또 만들어 지는 장바로와 도탱고 코튼 데시 잡아대가 속에 의절고 무가나야 도탱라 직다바로와 데스탄발라야 아니 땅포텔라야 가사 응컴는 장샤주게 도탱고게 셰샤테 개성아 속에 의절고게 도탱고게 셰샤데 스탄발라 데스 키요아지고로 코 디스 만도네 만도라 키야델라 팽그레스.
So here is an image, and this is an image that I like to come back to continuously, just so that we can sort of place ourselves and know which section of this living staircase we are learning about at any given time.
다 단다게 델라야 리모치에스타 리모데 델라야 텐치 ngachude 양시 직다게 양시 다야게 준생가렌 시스템발라야 ngachu고 드드치야게 조시데 단다 리모딜을 해봐 따바임바나 리모데게 차시 가와치를 핸도 델라야 ngachu게 콘트게 다 델라야 지차시가게 체도 인델라 양시 다이스.
And today we are going to talk about embryonic development, starting with only a single cell, and then we'll talk about how that grows and divides and creates its own different tissues, which are all part of organ systems. And then ultimately we'll get to talking about having the fully developed organism that would be born.
다 데랭아 속에 트야데 소데 갈라나 타폰 치니 제 구츠게데 타폰 치궁우 데라타 되게 제라야 네 타폰이 폰트블라 간다 드고요 바다 네 폰트라 텐데다 르보가 말라체 네 타마텔라야 징위 간자 치라야 간다 서버 치고요 바텔라타인스.
This is an image that I already shared with you on our first day of class. And I wanted to come back to it because actually Rickson brought up this point that he said, oh, okay, well, we saw this of when it was 6 months before you were born. So we know we have this picture and we know that you were born, but what happens in between? And that's what we'll talk about today, is that actual development process.
다 다들 퍼시탄샤이스 퍼티 개성어마 초던 담보값으니만 담보들 텐샤이스와 다 델라야 저게 린즈라게 트와치고 손스타 디 퍼디 임바이나 코 choram machi ge ngengirwa dawa tik kapsu ja chu pardere starwa ta dawa machi ge ngong ge dawa tik ja chu parde ta ani chi ge parla ya kare chong yo re sa twa ko sonsta tin sta mala ngasu ge tera ne machi ge ngong ge dawa tita karang la ya ani khola ya ko kanada uchi chongu yo metin de ja ins.
We have a little timeline here just so that we can know where we are in the process of development. Over here is where we have fertilization. This picture is me at 3 months, and then all the way over there is birth. So each of these represents 1 month. All of the lines represents 1 month.
다 텔라 타바임바나 로 리모데 티데타 가 텔라 ngasu thite ku tsugi ta kanado chingoryo metereswa ta phajo tambote sabun dijorwa sabun dijor deres ta parde jagaite dawasum gornu jila sabun dijor chi dawasum gornu jila jechu parchreswa ne susula ya thala tabaim bana de ta chiya gitu sutir to.
As I mentioned, we are going to talk about two different ways of looking at embryogenesis. And the first one that we look at is really going to be focused on these first 3, maybe 4 weeks of development. And one of the reasons I want us to focus in on that is that these early stages of development are very, very similar across all kinds of organisms. And we'll look at some examples of that later.
Ta tirang dina nga su ngu to chiagi chiti karin system balaya ane ko sabun dijur chinu ge jelagi ta dunta sarper ge dunta thoma sumni shi ge paro telaya ani karira uchi chungu yo mete tagi ins tate ta nu ge junsen karin system balaya ani thomi ge dunta sumni shi ge rang la sarper ge druagi neta chiwarwa te chungi mampu chila ani thmon to yo pachires thmonto yopachi inza jingu senada sochas gorwa ko thmonto yopachi inza ani telanga suge tampote mixker de tantagi ins thoma gi dun trasumni shig baradala.
All right, so we will start with talking about fertilization. And this is an actual microscope image of where we have the egg and then the sperm are surrounding it, and this is the time when fertilization would occur.
That dikarana nga suge sabun rijur gerbuji che phajotela ya dikarasena nga suge thaton chishir ola ya jeju parchi yes rata ko phajok kermo chewate ani nga suge khamerje rwa ani tig gangla yopate khamkar germo khashidwa otete thumigi ani kapki partires.
And I'll note just a few of the sort of most important features of this image. So one of those is that it's saying the first step here is that there are going to be multiple sperm that are contacting this one egg, or the oocyte is the word used here.
Ta nena ne kuge kasugure ta chechu ke chembu gnane tampote kare shigure sena ne de la oocyte side ta ngasugo gondor wa de khamer wa ye khamer mi jamba chigre ta di kasugure sena thomate la ya khamer la ya ani thuni ge kham karte tampo mambu chi yara khamer la tku yores.
And then even though there's multiple here on the outside, it's going to be only one of the sperm actually gets to the inside here that we see in the center.
Ta de la ya kula tampote la ya khamer la thuni ge kham karte mambu yo paim ba na de na ne chi khuna sugro ta kham kar chi khuna te khamer na jut ke yores.
So if we go back and we're thinking about what we know about genetics, why is it important that we have only one of the sperm nuclei or nucleus get into here? So we already have the nucleus here from the egg. Why is it important that only one and not a whole bunch get inside?
Tandane kasanga su rigzi rigpa kongla samne tang ba imbai na aswa rigzi rigpa kongla samne tang ba imbai na ngasu ge khamer gi nin tinin te kongla dwarwa tandane ngasu ge khamkar ge tinin chi te na ju ge dwa re mare rigzi rigpa kongla samne tang ba imbai na mampuchi manjwa chi khamkar ge dido kas tinin mampuchi ju ma thpa chi kangchin ju nu ge jinsen ga re sa.
Din sir, din mangbo yoena ni pi taksho yaksho di roena ani jini pu di ani yakbo jikke mega ke jora. Modi.
Nga ngi ko che na jamba de ne ba zo do wala.
Kamar chi ro, kamar.
Ne ba zo do wala sike. Kenda.
So we are hearing a lot of answers. One is to have the best offspring, and some are saying like we have only one ovum or one egg, so we have only one sperm that can fertilize it. There's only one, and some are saying the other sperms, their energy is lost, they're exhausted.
Okay, so I think from those, I want to talk a little bit more about their responses, saying that we have so only one from the egg and one from the sperm. So let's talk about that a little bit more, because I think that's getting us in the right direction of what we need to talk about.
Tanda len mampu nang deshas len mampu nani chiki kasigudu sena ani khamer nala khamer ge tinin chi orwa hotel ya kamkar ge tinin chi dus de go re sa ge lunch mambu chi na de shas ta tige ko nga su drusa ge khasho te la ya tata chi thik de shas tin sta bala nga su ge kamkar ge tinin chi ta kamar ge tinin chi orwa oti nyam de go re sa te tagins.
So if we're thinking about how many chromosomes humans have, do you remember that? We looked at those pictures of the chromosomes on the first day. So how many chromosomes do humans have?
Nilaya chopon katsayo res. 46 or 23 pairs.
Yeah, exactly. Okay, so we've got 46 in total, which means 23 pairs. And so where would those be coming from? Right? So how many chromosomes are we going to have here if we think about the egg?
Ta tanda nga su kamera la samla tang pa ma kamera ga na la ya chupon ka che res. 23.
Okay, yes. How about the sperm?
Kamkar la chana 23.
Okay. Yeah, so in this sense, right, is that we're going to need because we have the 23 pairs, and so we need for each of those one to come from mom and one to come from dad.
Ta tanda de nga su la chirgin ba na nyusasun chagu yorwa cha nyusasun gu yorwa cha nyusasun stambala ya ane nyusasun te phani yorwa che nyusasun te mani yorwa res.
And so that it's in this sperm nucleus, that's where those chromosomes are coming from. So if we had a bunch getting in there, that would be too much. They wouldn't be pairing up how we need them to for correct human development.
Ta michi cha la ya nga su te guya te cha nyusasun gu yorwa cha nyusasun te phani ane nyusasun tang mani nyusasun stambala da cha nyusasun ta da cha gu yorwa ti mai ba chi de te ke te nga su la khamkar ma tho chu chinan zu bai bai na ane ko cha ta te dga yong gu yo mares.
Yeah, so after fertilization occurs, now we will have one cell, and then it has the full set of DNA that it's going to need to start to divide and to begin developing.
Ta tanda nga su sabun drijo chinu ge je la ya nga su thapong kang chi tuk yo res thapong kang chi trip pa la ya ane ko la ya chopong cha ni sasum cha de ton chu thapong ta chi cha go re cha song chi cha go res ta cha ba de ane thapong te cha gu chi de te ane mang ta nga su ge da kang sa chi cha chi ro wa o te cha ngu chi yo res.
All right, here, this is going to be just the big picture overview of 3 main stages of embryonic development. And after I go through this list, we'll look at each one in a little bit more detail.
Ta tanda de la yo pa de nga su kari re se na dumsing gi charpe garam ba de res ta ko de se na ta ko thapong chi cha cha so sabon research ji ro ta ti gi thoma gi charpe garam ba de res charpe garam ba de la ya rumba sum yo res ro rumba sum di tanda dus chi che de la tu ko shayos ta de je la nga su rumba re la ya kari chung yo meta ins.
The first one of these is called cleavage, and these are just the very first few cell divisions that occur. So we're starting with only one cell. Of course, we need to have more. So the first few cell divisions are called cleavage.
Ta del tampo de la ya samjir se gores ta samjir se di kapsu kari chi gores na tampo te thapong kang chi oro ta sabon digong chi oro wa thapong kang chi puti thome gi chala ya thapong khashi chi la jiwa garam ba chi oro wa de la samjir se gores.
And then the second stage is called gastrulation, and this is where there's going to be a formation of 3 distinct germ layers, and I have definitions for those on another slide.
Ta nibade kares na nga chu dodrum driven size to ati ga ramba de cha gores ta di kares tamala ya nga chu ge dumten na de kore ta thoma ge thapong ge kola ya ramba sum chi tag do wa ramba sum chi tra chi gores ramba sum tribi ge ane ramba de res.
This afternoon we'll be creating 3D models from clay for these first 2 stages so that you can actually get a better visual in 3 dimensions of what's going on for these 2 stages.
Ta tiran yang kon jab la im ba na nga chu ge te kar sa ta number pe che tang de te thoma ge ramba ni shi ro wa ramba ni ti ge ane pezu zo yin sro shok cha sim deng ge pezu zo yin sta te zu ge kap sa ne che so la ya thoma ge ramba de ni ge kap sa kare chi jong go yo pa la ya ane ko tok te sa u yong res.
And then the third stage is neurulation, and this is the formation of early organ systems. This one is very complex to model, so it won't be included in our model, but we are going to talk about some of the genes that are important for these stages.
Ta sumbati karesena wangsa ke ten sigdo wangten dupigar batires ra tati karesena thuma ge ta thuma telanga su ge wambu ge mala sigoro ta wambu ge mala te chaga garam ba chigores ra ta didi sumbati bana ngasu ge chikarsu piz ge zu de ta choya tes khapu chigres dis tambala ya ngasu ge ya den deshe jagis.
All right, so here, I think before I get into the details of this, I'll just note the presentation of visuals here for learning about teaching technique purposes. So here we have the drawings of what is going on, and then they're all matched up with these microscope images. So the goal here is to be able to look at the same thing in these two different ways, with the drawing and with the actual picture taken from a microscope.
Ta tanda de se desham ko ge kari imbate deshe majong kola dusta ba imbana gangati ka res stambala ya ko ngasu gita mi ge lapa ga remote sa ju res ra ko imbate na kare kare jongo yome de remote sa ju res ta te ge okati ko thaton chise ge ola ngue sta per jap ju res ta tanda de ko se imbana ko remote ta ba imbana remote sa ju ti tam per jap ju te sha ko ro doas.
Ultimately, in this stage that's called cleavage, there is going to be that single cell that we've already talked about, and it's dividing by mitosis. This is why we had the brief review of mitosis, to form this hollow ball that will have about 1,000 cells.
Ta tampo de la ngasu sumju se di ka reses nambala ya ro ane ga te zupon ni chi rambala tene de te zupon ni chi rambala tene de te ane thapon chagu chi res ra ta tis ta bala thapon chagu chi ne kar chi res na ta ko re ta polu go re ra chi chag res ta de la ya de dina do chi cha pick up so ko na la ya thapon halam chitong jam si res na la.
I know yesterday there were some questions about how long do some of these processes actually take, and so this is different from plants, but there's a little timeline here showing that to go from 1 cell to 2 cells, this will take about 30 hours, and then to go from the 2 to 4 cells, that takes about another 30 hours for 60 hours total.
다 계산 바나야 제수가 나나 타폰 차고 집이 값을 름바 소슬라야 아니 치소가 선거 그래서 딘다 지기 되셔 개체 지기 되셔스라 데데 응아수고 저신가 타폰던 다 말이스 임바이나야 데라야 가수 로미기 다 타폰고 담 토미 차라야 차고 지야 들어와서 어딜라야 사운 디고네 코 차고 지야라 따바 임바이나로와 아니 토미 타폰 찍네 타폰이라야 차고 지야라 소숨치 탐바지 And
then the entire process to go from this zygote, just that first initial cell, to go all the way to the blastula stage, this will take 6 days.
Blastula system patient yudum in some sort. You drink the duelaya and you might drink yours.
And there's something really interesting about this stage of development that you actually can tell from looking at these pictures. So we keep getting more and more cells, right? We have 1, 2, 4. But I'm going to ask you, what do you notice about the overall size of this entire area?
Sarpaga Rambadilla Tapana Tampo Tapon Chigitane, Yitane, Shitane, Mampuchila Dribi Capsu, Kogi, Chichon La Tapana, Chichon Karatongodus.
Yeah, exactly. I understand Chikpa. So yeah, exactly. They're the same. So at least to me, I think this is such an interesting part of this process because the cells keep on dividing, but the individual cells just are getting smaller and smaller, so they're taking up only about the same amount of space.
Tade ko chechonte chikpa de gores ta de kapse yang ko yan sambuchi karyo system balai thapon shine thapon mambola ta chaguchi gorwa mambola chaguchi gore chaguchi kas thapon sama chont ro de ko chagu mampochi cheba imbai na ya ta ko tampo yo patan thama yo ko sacha se gi chechonte chibar chagdoa.
All right, so this is the general overview of cleavage, and now we're going to start taking a closer look at gastrulation.
Da did kare system balati gala tempate ngasuge sumjurgi rambago te Canada uchi meteres ta tanda imbana ti chela ngasu dodrum tin rupi rambatela tains.
So after cleavage is complete, so that takes about 6 days, then we'll be at the stage of gastrulation. And here we have what was originally just a single layer of cells is going to get reorganized into this 3-layer structure.
Ta ngasu sumjur te la ya thome gi nima tukurang la chong do arwa ta nima tuk jela ya chong te dodrum saetres ta dodrum saete karchi goresena de kapsa karchi goresena tampo te la ngasu la ya thapung ko rimba kanchi segora ta ko tepso jai ga rimba kanchi yo pate ane ko rang chedri chi de tang rimba sum yo pi ngu chi lan jigres.
And here, I'm just going to point out, so I have only text on this slide, right? So right now, it's probably a little bit difficult for you when you're thinking about, you can kind of imagine, right? You're like, okay, it's 1 layer and we're going to 3 layers, but it's a little bit difficult to know exactly what is going on here. There's only text.
Ta tanda de kapse chesula ya ndela tembati na yigi masio maris per yo marwas ta deshe karja gores na rimba chi yo pachina rimba sum yo pachila ya ne ko jirgu yo resegdo as ta tin stambalaya ta rimba chine rimba simlai jirgu sag yo da imba na tong is kanaru chin jirgu yo pa na kare chin jirgu yo pa sa te ngasu samlo na cha khapu as.
This slide also only has text, but I promise the one after this does have the image.
Tan dela yan tsichipu yes imba de javla ya ne remotengies.
Here, I just wanted to make sure that we have the vocabulary for these different terms. The ectoderm, the mesoderm, and the endoderm.
Dako pontu rumba sumchi orwa tanda dela ya ko rumba sumi yo patela ya ne chishin tang person tang nangshin segres ta de yang ko kare yimba te tskiti ngutu cheg che de ten yes.
So as we can see here, the ectoderm, that is going to be the initial starting cells for the nervous system, for hair, and for skin.
Ta chishin saate tamputa ko rumba sumla triggerwa chishin saate ane ko ta rimsin te serper chi ko kare la ndukure na ngasu wansa malata ta ta puta ta ne papa lanjur grace.
And then the mesoderm, those are the initial starting cells for muscle and connective tissue.
Ta person te ngasu shadrim ta thujur fontala ya chagayores.
Finally, the endoderm, this is going to form sort of everything for our intestines, and then also the lungs, and actually several other organs as well.
Ta tiga ola ya nangshin sa sto nangshin te tambo ngasu ge ta tuje bulam sa stambala khala jasa ge chigo ta ngasu ge jimala sopa de che ti senda ngasu luwa che ni senda ngasu ge nala yo pi ge wampo manchi shi chi tukuyores.
All right, so here is the image that I was mentioning, and hopefully this will be really helpful in showing these 3 distinct germ layers of the tissue that are forming.
Ta dite ta remote yens ta ngasu ge ko ge drums ka sa nyu drum ge ramba sum trach gorwa ramba sum ti trya ge ne remote de ten sa yos ta dela ta chi de te che chu ge ta ko ramba sum ti kandast ko yo ma kar kar ye me te ya chi te sa u chari wa chi ge yes.
Here I'll just point out that in the previous slide for the endoderm, we sort of just said, oh, okay, and some other organs as well. This one provides a little bit more detail that the endoderm will eventually form the liver and the pancreas also.
Ta tangi tinala ngasu ge tikasa nangshin ti imba na ro nangshin ti la ten de ta ngasu ge wampo khashi tukre se gorwa ta dinala ya ko khasu ju sha liver se na kar chimbarwa ane pancreas la na shermarwa otte tukre se dekha se utshas.
And I wanted to make note of that because we are going to look at a few of the specific genes that are involved in that process later so that you can have a better understanding of exactly what are the molecules that are needed to create those tissues.
Tanda de kasuga suga chimba tanda Sherma said the Desha de la Chituna Chiro Chase, Ra, that the Jela Yangasu Karila Tagresena. chetra ki wambo deta trivia la ya trivia tan joyo pigi rigzi la ta gi insra rigzi chira bala ta insta bala ya ale rigzi de imbana ne wambo deta tripku dua se ge turji chit kyores de la to no chis.
In the first image here, you can see this looks very similar to that image that we ended up with at the end of cleavage. So here we have those cells that were up at the top in the previous picture. Those are called the inner cell mass, and those are beginning to differentiate. So that means that they are beginning to become more specialized kinds of cells.
Ta tanda do sta ba imbana remo tampo de tanga nga chu sumjir ge remo thama te tan rawo yore swa ta de na ne thoma te lai inner cell mass do anang ge thapong ko chopa se sto ta de kap se de kare se tambala tampo ko ra chenju chi yo mares parwa ko chenju chi yo mares ta tanda de kap se ko chenju chi la ya tari chi yo pan uchres.
Another thing to note about differentiation is that before this occurs, those cells have the possibility of becoming any of the cells in the body, and it's only as they go along the path of different kinds of signals that those cells are receiving that they will become more and more specialized into all of the organ systems that we know about.
Ta tanda de kapsiya shende chi karingi gure sana khutsu chenjur machi ge ngö la ya de chenjur machi ngön gi thapon de talas thapon de tala ko zupu ge chashi kari imbana drupi gi nibati yores ra iman ta ko de kapse ta ko chenjur chi ang go zupa dan sagro ta ta ge jurum de la dro ta ane ko la ya ta chi ga da ta chi ro ta chache mampo chi la ten de ta ne ko chenjur chi de ta ta ko thapon mandang wo la dusun jurgu yores.
Over here, this is where we can actually see those three layers that we were mentioning. So the green that's sort of on the top there, that's the ectoderm. In the middle, we have this little bit of pink, which is the mesoderm. And then down here on the bottom side in yellow is the endoderm.
Ta tanda ge rumu thamadela ta ba imbana nga chuge ta khuga rumba sambudi ha ku du sra rumba sambudi kana sa kundu sna thoma ge jangkho yosate chishun sayati chirwa ane paro ge di person sayati che ok sarse yosate kare sistamala ya nashin deres.
And then the main point that I want to make about how this process is occurring is that in this region here, there's a lot of cell movement, a lot of cell migration that's happening. And by all those cells moving, they move like this. That's what's able to create these three layers.
Ne ko ta rimba sumtin drivya la sigoro da kandas trikresena tanda dig kapsa ne ko thapung ge tang gucho sigoro da cell movement thapung tang gucho shi chambu chi ge yores gucho shi chambu tila tin dete ane ko ga rimba di mando sumtin rit ge yores
chiwas tan chiwas sesha
one caution
Yeah.
tanda ge ge disunsa tanda tambo dine niba tambo dine ko la chabu chama la ko chenju che ge niba yer la sa ni tijilo so different yeshing on judugda til da mombo ji le ndam da juji chogolas ti da da so khawani jogo resta chorong gocha chora kamba che chora laba che sene ta da ji jogo ra ya di da da so khawani jogo yo resti
so from the first picture to the second picture before the cell differentiation you have mentioned that the cell will differentiate due to the different signals they get. So my question is, where do the signals come from?
Yes, I think this is an excellent question, and I think it is the exact question that everyone is always asking themselves, because I think what your question is getting at is kind of what's the very first original signal of where these are coming from. And some of this is still being explored, but I will say one of the signals that I do know about is that some of this is related to the positioning of where the cells are in relation to each other. I think you could still make the argument, how do they know that? How do they know where they are in relation to each other? But that's kind of as far back, I think, as we have the knowledge right now, is about the positioning of the cells.
다 단대 차라야 안에 그 드라티카레 시스템 발라고 다 만푸치라 텐데 단 그 타폼 만동우라 천시 지고 세고레스 네 다티 코는 종레스 드라티레스 다 팀바나 다 드라티 만푸 당도 양시 용고도 다 소티 타마텔라야 다고 딘다 천주 지야라야 다 토마게 다티 코네 용고레스 다티 단도 하고요 말레스 다 하고요 말때 독시 지단 지신 바레스 다 데라야 대세 자가게 탑는지 가는 대세 자고레스나 아니 타폰테 딱가 네살라 스고라다 고수고 뒤지게 네살라 때네 스고라다 고가 네살라 때네 데티 아니 고 다만도 용고레 스고레스 임바나 데타노 고 카세오고네 데세자트키오 마레스 단다이마나 탁시 치탄 치진 마레스 지게 고가 네살라 때네 데테 네 타폰데 델란저 군두 사치고라 다가다테 용고레스 데세자고레스.
And we are going to talk about another environmental factor that influences the signaling in reptiles a little bit later today. I'm not sure that it's so early in development as this, but it still is a similar idea, a similar concept.
다 데랭아스 제라야와 데 둔리소 초치요와 둔리소 초게 나라야 양코 코이글라 텐데 데타야 코다치 용아치고레스와 코이글라 템비 다 용아치요레스 따팅아 수게 제라야 데세자인스 임바이나야 코이글라 템비게 단루야데 단다게 양코 철페가 름반 디기 강라 용고요메 텐데 하쿠마소 임바이나 둔리소 초가나라 코이글라 템비게 다 용고요 바테 젤렌 데세자인스 Can we call that a chemotaxi?
I think that we probably could. I mean, there's definitely some signaling going on there. So I would say it's, there's not necessarily, from an external source some chemicals, but the cells to each other are signaling and then moving. So I think that we could think about it that way.
Chemotaxin sigdoangasu zelatene guchotanga chorwata ko tela zeda teletene chongala dikmarwes te kapsa chichi bana chalone mai bana ya ko chapong kutsu bana gucho chongo arwa chapong kutsu gucho chongo re gucho chongatene chapong kutsu ko pansala ya ko da tege arwa ya ko gucho tembe gidasena la digres gucho tembe zidasena
tanda de tambojidage do sama stem cell res tanda de ramba tambonade
So in the first picture, all the cells in the first picture stem cells? Are they stem cells?
Are they stem cells? Yeah, the ones that are stem cells are up here in the inner cell mass. So it would be the cells that are part of the inner cell mass. Those are stem cells, yes.
Then the stem cell is seperti the inner mass cells itu angasu nangi tapung coba saya itu kan. Hotina na yo pergi tapung sama de makan tapung cagiores.
Kita di gandelara the fertilization chazani del jargod la du tsamba ti bara thuju gyuring le khaju sena na
kare
fertilization ne jargod la pa du tsamba ti bara thuju gyuring le khaju sena.
So my question is how long it takes from the fertilization to become a zygote?
That's a good question. I think some of that is a matter of definitions. I think that usually we would just say as soon as the egg is actually fertilized, then we would say, okay, now this is a zygote.
Singatsukarsa tate sabun region ne sabun digun chaya te sigorwata ra ate chaya la te so gazu gogre se te yanko ko chenji shatan chila rali gores ra Kuton dresse jatan chila rale gores kashi imbaina ko fertilization sane sabun digu yorota sabun di bata ane sabun digon te trip research yores.
And it's possible, I don't know if you're thinking anything about, like consciousness, something like that. That's not involved in this definition, and I think that I don't have the answer to, but just in terms of this terminology, that's how it would work.
Tanda chingi ki serkanane ko tanda chenji shantan ti shisha bana ko taran chago res sabon dijo chiwa ta sabon di kon trup ko yores ro chichi bana ya cherang go samle tanta te sabon dijo chiwa tila tende dete ane sabon di kon trup ta tinan tele ta ching nam shishig ma shik samle tang yo bana tanda ki ko ge chenji dinala nam shishig ma shik tila ya ane ko shisha yo mares do yo mares.
I think so. No.
The dinala di doge.
Taji Tangjim Balajda.
Ah.
Taji Tangjim. Cytoplasmic determinant. Kogi then the Dichigio Marivil, the Pena Trapung the Kaga Kagi Pena, Gocha, Kamacha, Lavacha, Chigura, Chagocha Jura, Kogi Chigio Marivis.
So my question is, in this one we have a cytoplasmic determinant. So
A cytoplasmic determinant? Okay.
So will that signals the cells to differentiate in different part of the body?
Okay, so are you asking if it's something within the cytoplasm that's going to signal the cells to differentiate?
Yes.
Yeah, so I do think that I would say that is true because so we have the DNA in the nucleus, and then it's eventually going to be made into, or have the RNA copy, and then the proteins will be out in the cytoplasm. So I would agree with that. Yeah, I would say that there's some proteins in the cytoplasm that are, or in some other maybe peptides as well, but things in the cytoplasm are doing some signaling, yes.
That de la yan tanzania te gele so ba na tsi tanzimba sa chi yo res ta ti change lo ya shi chen tugure tuguma res ro ta te chi chen de samna tang ba na tugure res nga so DNA te tang po ta tin na na yor da imba na ko phapsi chi ka sa phapsi chi ti je la ya kwa ra ne te nga so thachi na yong go yo ro Otela tene de te yang thachi ge na la ya tangzi chi ta thachi nang ge chi na la kari yo me la so pa la tene de te yang ko change tu la ya chapter ta da ta te yong res
Was there another question back here?
Yes, Anamala.
Tanda bar thangmudne amnion lashto akla. Ondi ngonzo bugunzo bu puyin pomoyin tadu dine dis challenge bu puire pomore hagu da. Ondi nalola ni hagoela kari yores.
So my question is, sometimes people take a cell from the amnion from the part of amnion. Yes.
Okay.
To decide the gender of the child. So they can just figure it. So how we know from the cell from the amnion?
Yeah, so
gender of the child.
Yeah, I think that if I'm thinking about the same process, usually that is done slightly later in development. So it probably, I mean, this would be, I mean, right now, this is still, as it mentioned, only about 1,000 cells. So to be able to take a sample of that would be extremely difficult. So you have to wait a little bit later in development. But then, yes, you can take the sample. And so that's even, like the chromosomes that I showed you that were mine, that's how those were obtained. They were able to take the sample from those a little bit later in development. And then that's how they could tell that I had the 2X chromosomes.
Ta tanda de nga su ge amnion sa de ngel chi sa tro ta nga su ge nge chha ge ni dung ge pa pa ge tirwa cholo la yo ga tirwa ta de khu ge sa la ya te na ne thapon chi len ti thase chi bal te de ta pho ta mo kare ba ha ku dus ta tanda ge ni kap de la ya nu ko ha ko khabu chi ge res ta na de ka sim ba nga su thapon ton tha chi sma shi yo ma ro wa ba na ya tanda nga su ge nge chi ge na ne ko thapon len de ta ti na na yo pi ge ris ko ta nga su ge 최폰때 안에 카탐 데이터 따고 여러 와타티가나 XY요 XX요 데 따고 여러 와 데데용 코 설프가 럼바티스 치스레스다 제라 kana kanse separate chonu ge jela ra ma to tangbo de la ya ko ta kha pu chi ge tha pha chon nyon ro.
Thank you. Here I wanted to do this just a brief review of what we just talked about before I then go through the second way of looking at embryonic development.
Ta tanda de la ya tanda ge ka sa ta adrimsang ge chapter ge ta ta ni yo re song song tangbo de wa ta nyo te ma ti ge ngwe la yas ane tangbo ti ge na la ya karen de shi ja pa ti ge ane ko pi song ge mu chi ins de.
Okay, yeah, I realize some of it's a little blurred, but that's good. So here from the beginning, we're able to see those initial steps that we talked about called cleavage, where we have just the one cell going to 2 to 4 to 8, and then eventually down in this area forming the blastocyst.
Ta tanda de kasro nga chu ge simjir ge kapse ko keche she thapon chigine ni ta shi ta tane chik ton la de la so pa yorwa ni tanda man de yok pa ba nga chu ge tikasa nyudrum drung ge ni tan te lagi yores.
And then after that, we have gastrulation, and you can see they have the colors for the different cell layers here as well. You may be able to see this a little better on your own screens, but you can actually see a little bit more of how those cells are actually moving if you follow each individual color.
다 되게 제라야 가지고 도든 게 럼바티 연구요레스 거기 차풍 그쪽도 차풍 차규지었다 네타티 간자 데라야 지연 천천만다 만다고라야 스탠샤요레스라 아니 단데라야 지야포 지 통도코 통고민도 스팀바이나 체수게 로레간라 타바임바나 탄유치 타바임바나 쳔보조치 타바임바나 아니 거기 차풍 그쪽도 차풍 천천만다 마라요 바투조 응원스도 부통그레스.
And then that takes us to that third stage we mentioned, the nergulation, that will be starting over here. And then as you can kind of tell from this, it's going to become much more complex. And then here, this is an example of human, so it's actually forming that human baby.
ko rumba sumcha ge ne tante yang do at ne ta rum sin tan man control ba bana ne ga su ge ta ka sa mi gi chipa sa go ta mi gi chipa cha ge te ngo so do bo ne thong do as rumba te
There we go. Okay, so now, and the second way of looking at embryogenesis, it is a little bit shorter. It doesn't have quite so many steps and details. And I just want to let you know for this one that it actually is going to apply to mostly the whole time frame of embryogenesis. So this one won't be as specific to just the first month.
Dontra ninis kas sumni shige parola ya ngose to pakario mete ne tante dese jage do arwa ta tanda ge de la ya drumson drum de la imba na khoge chiga thome ge ramba de la ya cheta ka ta de na yo mare state ko kanza ge ramba chila ya nyam ta gres.
This picture is just here as a reminder to let you know that these steps are still relating to the same process. It's not talking about a different process or anything like that. This is still all embryogenesis.
Da de bana tando bana ngasu go dum song drum res ta de la ya rumuti to che ta tande ge dese jatan chi bana ro dum song drum te ge na la ya ane rambasmi yo pate teng yores.
This way of thinking about embryogenesis is going to be really focused on what are the cellular activities. So this is thinking about like, what are each of the cells doing as embryogenesis occurs.
Da ko dum sang drum di jombe kaps zo de chapong gi jeli kari chi gi yores ro da chapong su su gi lika kari chi gi yores ti shi sha chi de tan de se ja gi yores.
There are three main cellular activities, and we have talked about them. And like I said, this is just another way of viewing the process. So those cell activities are cell division, cell migration or cell movement, and then the cell differentiation.
Da thapong ge ko ge karsa jele ti imba na ro jele ti la ya ane ngasu ta che res ro ta de kon dese ja ta man ro chi yores ta tampo te la ya ne ko thapong te ta kar chi re tampo chagi chi yores. Anibatelaya ko ni puchi yores sacha jenda chilen rogo yores sambatela imbane na changer chi yores.
And the final point that I wanted to make about this before I give you all a discussion prompt is that even though these are numbered as 1, 2, and 3, at most of these stages of development, these will all be happening at the same time. So it's not that only one happens, it's complete, go to number 2. These can all happen at the same time during embryogenesis.
Tandagi Nirmalaya, remember the Sumchachi Tampun de Chon, Titsan de Chon, Tigilan de Chon Sayo, Yamton Jongores, to Yamton Jongores.
As we saw in that learning objective, one of the big objectives for this morning is for you all to compare and contrast these two different ways of looking at embryogenesis. So I would encourage you to discuss that generally. And in case it's helpful, one specific thing that will be really helpful to think about is for each of these, see if there is one of the embryonic stages from that first way that we talked about. see if there's ones where you see that they're most related, that you think some of these cellular activities will really be going on during each of those embryonic stages. So we talked about cleavage, we talked about gastrulation, and we talked about neurulation.
다 신데데 카레 시스템 발에 다 단다 데 값nga su tampo 시중 거 무예 데세 잡이 값 시중 거 무예 나네 단다 데세 자타니 치도가 데세 자타니 때 총들지기 인사지고 에스다 체수가 단다 니 따로지스로 그런자 데세 자탄 데다 데세 자타 tampo 지어로 then 이거 바로라야 드와 카레카레치도 총차 카레도 총사 카레도 스테 따로지스 아니 체트게 가리 따로치에서는 내라야 rumba 숨친 데세자고도 와라 그게 젤리 숨친 데세자고도 와 제구 만도 숨친 데세자고도 와 제구 만도한디 tampuga nana deseja batige kapse yana sumjir ta dewa sumjir kasin jongdwe ti olaya todun debren kasin jongdwe yana wangten termen ses toa ti kapsin jongdwe kangi tan dewa dus ta dewa dus stambalan drive chashi chuandus tela taroches.
All right, so take a few minutes, discuss, and we'll come around, and then we'll hear what your ideas are afterwards.
나하다 가마 카시 치거랑라야 네 되든 나로치에사 엔티게 제랑아 소냠 타기에사 아니 쿠마데마시 페사 저런 뭐 제때 뜨는 니크르다가 드는 니크니 주드다 सामने तंग पाई बना तंदे चुरुंग के सबते तगारां जे दिगदुस राने गासु तंदे दे मांजे शादेता इन दे गाला या जाचे याला छाछ तंगिस ता जेंदा चिक सोंदा या
ngazo tes tada ginlos tamba la den rodo ji ji ani tes mando de khardala na ni poya je do aha tombo de ji ba ji ani ni basmani de ani jeno je gana tada digdos ni poya de ani ngazo tada gong shi ani na hadu ju ombo mala chabas ji do
ani poyate tanda de kasa nipu sate jwa ta gudo la gwa
oya aha tanni poya sate tangi kon hiwa ina ani rumba sumba de kon le ya siro aha ani tada rumba sumba dan tenan ka sir mani sumba je do sumba de tangi kon hiwa rumba ni bali ja ina ani paje tada ndik do sans dos
aha las so we have a different view on this for this case if we put like the migration into number 3 and then a differentiation into number 2, so we can match that three activities directly with the 3 things that we discussed earlier, the cleavage and the 3 of them. So we can directly match them. So in this case, they're saying that if you put the migration to the number 3, that
okay.
the migration can be directly related to the neurulation.
I see. Okay, yeah, so this is a really good way of thinking about it. And then, so I think now for clarity, I'm just going to go through and make sure that we know exactly which cellular activities are happening in each of those 3 stages that we talked about in the first one.
That tanda ta ngasu de la samna tanda yago dus tanda ta ngasu kari samna kari la dese jagin sena kari khase jugin sena ane tanda de la ya thapong gi chegu mando sums dugo yarwa thapong gi chegu mando sum deta gangla ngasu nyude rimba mando sum sin dese jaswa rimba sambi gi kapse thapong gi chegu mando kari dugo yometik sekhazu yis.
So we started talking about cleavage. And so that is just these beginning stages here, and I guess coming to here. And so I think, as you all have already mentioned, during that stage, that is when it's cell division, and there's only cell division at that time.
Right. That chetsu dele dese jabana sire tampu dele ngasuke samjir chige netan dese jago andaga parta samjir du de kapse ane thapong che chegu karen dugore sena thapong chagui gitin dugore jeram din dugore.
And then during gastrulation, so that's the second one that we talked about. And this it's a little bit tricky, but actually there's all 3 of these occurring during gastrulation.
that rodent driven go capture wa and chetan go nyok putis chi yores rodent driven go capture bana thapong ke chegun chu samchan jong yores.
So the cells, they are still going to be dividing during gastrulation. Of course, there's migration, right? Migration, and that's kind of the defining feature of gastrulation. And also, we're starting to see just the very, very beginning stages of differentiation.
Ta rodent driven tiga kapse ta karichung gore sana tampote thapong chagu chigu yoras thapong chagu chigu yores niba telaya thapong nepi chigu yores ro. Nepu che se ti karasana rodent driven ge chechu suti ya nepu che adres thapong nepu chi yores tin jong yore tin najing ge thapong chenju che chi ro tin mangpo chi go man nyong chenju chi yores.
And finally, for neurulation, which is what we'll talk a little bit more about after the tea break, definitely all of these are happening during neurulation. And this is where even more and more differentiation will occur.
Da tege thama tenga chuga wanten driven size to wanten tripiga rumba tege na la ya ane data samchan jongo yores thapong ge shego samchan jongo re tege na ne labar ta tandage thapong changer chia chi ro ko chele chenten jura ge te shicham jongo yores ta tanda tege wanten driven tege ko la ya nga su soche person ge je la ya ta ju nde se jagins.
Yes, so thank you, and let's enjoy some tea.
To cheers and such a new knowledge.